Hello, everyone, and welcome to today's EIS Talk webinar, brought to you by the European Aquaculture Society. My name is Anna Viskovic, and I'm the Project and Program Manager at EIS, and I will be your moderator for this session. Whether you are a student, researcher, or industry professional, we are happy to have you join us today for this another insightful season in our EIS Talk series. Today's presentation will last 35 minutes, followed by a 15 minutes live Q&A. So please feel free to submit your questions in the chat at any point. We will get to them after the talk. As always, this webinar is being recorded, and the recording will be available on the EIS YouTube channel for 1 month. After that, it will be accessible only to EIS members via our website, where you can also find all past EIS Talk webinars and presentations, PDF dating back to 2019. If you are not yet a member, we encourage you to join. Students and retirees can register for free. Today's talk is titled Optimizing Feed Palatability in Aquaculture, Science-Driven Approaches. Palatability is more than just a taste in fish. It's a crucial sensory mechanism directly linked to growth, survival, and productivity. As aquafeeds continue to move away from traditional marine ingredients, the challenge of creating feeds that fish actually want to eat becomes even more pressing. In this talk, we will explore how fish perceive feed and how understanding these sensory responses can help us design more efficient and sustainable aquafeeds. We will look at how feed additives can enhance acceptance, reduce waste, and ultimately improve performance, even under stressful farming conditions. We are very pleased to welcome today's presenter, Dr. Sofia Morais, Innovation Aqua Team Leader at LUCTA. Dr. Morais brings 27 years of experience in aquaculture research across both academia and the feed additives industry. She holds a biology degree from University of Lisbon, an MSc in aquaculture from the University of Stirling, and PhD in aquaculture nutrition from University of Algarve. Her expertise includes fish nutrition, lipid metabolism, digestive physiology, and feed intake regulation. At LUCTA, a global leader in feed additives founded in 1949, Sofia leads research and development efforts aimed at science-based solutions for improving feed efficiency, palatability, and animal welfare in aquaculture. We are also happy to note that LUCTA is a corporate member of the EIS, and today's session is being presented as part of their membership benefit. So, without a further delay, please join me in welcoming Dr. Sofia Morais. Sofia, the floor is yours.
Well, thank you so much, Anna, for the introduction, and hello, everyone. So first of all, I have to thank the European Aquaculture Society for hosting this webinar, and of course to all of you for your time and interest in this topic. So here is the brief outline of this of the contents of this presentation. So we will start by reviewing some theoretical knowledge about what is palatability in vertebrates, but more specifically focusing on fish. And then we will talk about palatability enhancers, some of its practical applications, and I will also give you some examples of use to finally end up with some conclusions. But before moving on to the technical content of the presentation, I just want you to briefly introduce you to LUCTA. So LUCTA is a family-owned company that was founded 76 years ago in Barcelona, where the headquarters are still located. Nowadays, it's a multinational company. It has presence in the EMEA, Americas, and Asia-Pacific region. It has offices in 12 manufacturing sites in the countries which are shown here in the map, but of course it's also present with sales in about 100 countries through distributors. Its global teams include 1,300 people, which work across the 3 different business areas, which are flavors for food, fragrances, and feed additives, which of course will be the scope of today's presentation. Okay, so now let's talk about palatability and what is palatability. So according to one definition, palatability relates to the pleasantness of food and its ability to influence behavior, such as the positive drive to eat. It is linked to the sensory experience of taste, but is also affected by nutritional needs and individual preferences. So, oh, sorry. It's a problem here. So this definition associates quite closely with the neurophysiological view, which explains the regulation of food intake by an interplay between the hedonic and the homeostatic systems. So on the one side, we have the hedonic regulation, which associates with the pleasure of eating, which responds to sensory cues such as chemical, visual, and tactile signals, which would cause the release of dopamine, which then signals the reward areas in the brain. On the other hand, we have the homeostatic system, which responds to the energy, to the metabolic, status of the animal, which correlates with its physiological status, its health and stress status, as well as the environmental conditions. And this is more associated to regulating energy balance patterns. So it's well known in fish that homeostatic regulation is occurring in a similar, very similar way to vertebrate mammalians. In the case of the hedonic regulation, it has to be said that there's still a lot that is not known about this system, but practical knowledge indicates quite clearly that fish are indeed able of discriminating sensory cues and showing clear preferences for different types of foods. But there's also another view that is, I think it's quite important also to keep in mind, which I think is very complementary to the previous view of palatability. And this is more related to its actual function. So in this case, palatability is described as a mechanism that evolved to allow animals to recognize and evaluate the quality of their food. So it's a very well-known fact that fish require nutrients, not ingredients. But of course, nutrients or their sources need to be recognized as food. And in order to do this, animals have evolved sensory mechanisms to guide them in making appropriate choices. And fish are actually quite well developed in this sense, as you will see next. Sorry. Okay, here we go. So from the sensory systems that are guiding fish feeding behavior, chemosensory systems are the most important ones in this respect. So on one hand, we have the solitary chemosensory cells and the olfactory system, which these are long distance sensing systems which allow the animals to detect and orientate towards the source of foods. It should be recognized also that they are involved in many other important basic biological functions. Then we have taste, or the gustatory system, which is a short distance sensing system, which is what finally enables animals to make the final assessment of the quality of the feed and then make a decision on whether to ingest it or reject it. Importantly, this system is evolved only in feeding behavior. Okay, but there are also other very interesting aspects and even unique aspects within vertebrates in fish, which is the fact that taste buds, where we have the taste receptors, they have both an oral, but they can also have in fish an extra oral location. In terms of the oral taste buds, actually they look just anatomically speaking, they're just the same as in mammalian vertebrates. We can see here in this photo, this is a tongue with taste buds and the close-up of the taste buds of European eel. And this would look just like you and me, basically. But some fish species can also have taste buds all over surrounding their body surfaces in the areas mentioned here. And some species even have barbels, which are specialized taste organs or appendages, which are covered in these taste buds. So important characteristics is that we have the fish have the highest number of taste buds within vertebrates, and taste receptors have higher sensitivity to taste than in mammals. So these make fish models organisms for taste research. What is also quite, from my point of view, extremely interesting and less well known is that taste in vertebrates in general, including fish, continues after feed ingestion. And the reason for that is because we find taste receptors. Sorry again, this is playing off the. Here we go, sorry. So we find taste receptors in specialized cells, which are called enteroendocrine cells, and I'm showing one here. And in this case, we have, and these enteroendocrine cells are found scattered throughout the gastrointestinal tract from the beginning to the end. So in these cells, we have the taste receptors as shown here, facing on the surface, facing the lumen of the gastrointestinal tract, where they are sensing nutrients and chemicals as they are traveling along the gut. Once they are activated, so the activation of these taste receptors causes the release of different types of peptides, and the type of peptides that is released depend on the specific type of enteroendocrine cell, which is different along the gastrointestinal tract. So these peptides can be ghrelin, CCK, GLP-1, and PYY, just to give a few examples. So once they are released, they can enter the blood circulation where they will have endocrine functions. They can also activate the afferent vagal neurons, so basically the nerve endings that surround the whole gut cavity, or they can signal neighboring cells like the enterocytes. So each one of these peptides will have has different functions, but overall and collectively, they regulate processes such as satiety, gut transit, digestion, absorption, and metabolism. So we can say that overall, the action or the function of this gut sensing system is to enable the body to have an orchestrated response or coordinated response to diet composition, which leads to optimization of feeding efficiency. So to summarize, what inputs can control feed intake both at the central, so in the brain, as well as from the periphery of the body. So we have the first step, which is very short-lived, is the sensory input, as I mentioned. So this happens seconds to minutes. Dopamine is released, signaling the reward brain circuit, and the function of this input is to drive meal initiation and food choice. Then as soon as the food starts traveling along the gastrointestinal tract, we have gut sensing, which is, as I mentioned previously, responding to nutrients and food chemicals. This is more minute to hours time scale, so it has an effect during and between the meals. As I mentioned previously, effectors are gut hormones and vagal afferents, and what this system is doing is to control meal size, gastrointestinal motility, so how fast the food travels throughout the gastrointestinal tract, digestion, absorption, and even metabolism. And then finally, we have the homeostatic system. So this is an underlying long-term regulation between hours to weeks. So it is responding to metabolic and hormonal signals, which are circulating in the body through the circulation, managed by the regulated by hypothalamic centers to regulate energy balance and nutrient reserves. Okay, so now moving into palatability enhancers. So as you can imagine, in order to develop palatability enhancers, we first need to understand what stimulates fish senses and why. So from a chemical point of view, we know that chemosensory active substances, meaning ones that elicit attractability and palatability responses, they are always low molecular weight in water or ethanol soluble. And this is because they need to be dispersed through the water. They are present in high concentrations in preferred food items for aquatic organisms. And once they are released into the water, what they are doing is that they are signaling the presence of good quality food. We also know that there are important synergistic or additive interactions, that mixtures are always more stimulatory than single compounds, which of course makes sense because if they are signaling the presence of food, mixtures are always more specific than single components. Okay, but in aquaculture, we don't have praise. What we do have is inert feeds or aquafeeds. And in this case, what we need to consider is the composition of the ingredients that are used, more specifically, the soluble ingredients, how they are released into the water and how they are going to be sensed by our farmed animals. In this graph, we can see some typical levels of water-soluble compounds in ingredients, and we observe here chemicals which are considered chemosensory active. They are typically present in high concentrations in fish meal and marine ingredients, but not so much in plant ingredients. This graph is also quite well known for all of us, and it's basically showing how the level of inclusion of fish meal in aquafeeds for different types of species have been decreasing in the last years, and quite well pronouncedly in the last 2 decades. And not surprisingly, if based on what I've been explaining before, when we look at publications in Scopus, the number of publications which have fish, feeds, and palatability as keywords, we observe that in the same period, there's been a very dramatic increase in the number of these publications. So what this is telling us is that the interest in palatability is increasing, possibly because as fish levels are going down, we start to face more problems with palatability and feeding. So a way to mitigate some of these problems and bring these sensory cues back into the aquafeeds is to utilize sensory additives. Before giving some actual examples of this, I just want to quickly explain to you how in Lupta we have approached the development of this type of products. So we have designed 2, let's say, types of products which are under the Lupta-Rom and Lupta-Max brands. They're actually not that different from the point of view that they are mixtures of known chemoattractive substances, where we are targeting mostly, not only, but mostly taste receptors. So the Lupta-Rom type of products, these are liquid products which are intended to be applied through coating with the oil, preferably through vacuum coating, so that the objective here is that we have these chemoactive substances at the surface of the pellet, meaning that they will be quickly released to stimulate olfactory and oral taste receptors. And as we have seen, this type of sensory input has a strong effect on improving appetite and feeding. On the other hand, we've also developed other type of products within the Lupta-Max brand as powder versions to be applied in the mash. So what happens here is that there is a more, let's say, gradual release of these chemicals as the food is traveling through the gastrointestinal tract, since the objective here is to stimulate taste receptors in enteroendocrine cells, which activates gut sensing mechanisms, which again, I've explained before, has an effect on controlling satiety and gut function. Okay, now moving then to these practical applications and examples. So first of all, just a quick brainstorm on when is a good moment to use this type of palatability enhancers. Well, first of all, to overall to mitigate what could be key palatability challenges in aquaculture. So I've listed here some of the main ones. The first one is associated when to reduce levels of marine ingredients and high levels of plant-based ingredients or some novel ingredients which are somewhat unnatural for fish, so they don't normally found them in their normal feeding habitats. So these typically lack natural chemoattractive substances which reduce feed acceptance and feeding efficiency. In some cases, we can have beta compounds in the feeds. This can include medication or certain types of additives, which can lead to feed rejection or low consumption. So this causes wastage and pollution. Of course, management and especially environmental stress is on the uprising in recent years, and stress causes a strong negative impact in feed intake. And we should also consider that globally, there's a huge variability in the number of farmed species. They all have their own taste preferences, And at the same time, we have a limited number of ingredients that we can use. So this type of additives can help us tailor formulations to species taste preferences, which is particularly important in long domesticated species. Okay, now moving on to a first example of application. In this case, we tested a looked around type of product. If you remember, this is a liquid product that is applied through coating. And our objective was to test an effect on overcoming stress-induced anorexia in gilted sea bream. So we did this trial in collaboration with the Institute of Aquaculture Torre de la Sal here in Spain. And as they have a very interesting setup where the recirculation system tanks are equipped with demand or self-feeding systems, which basically are activated by the fish themselves. So the fish learn how to pull a sensor which delivers the feed. So feed intake is completely voluntary in this setup. So as commonly done, we have an acclimation period where the animals are fed their common commercial diet. And then we have two phases. An undisturbed, where initiating the experimental diets, and then we induce a stress, usually for a short amount of time, about 6 days. And in this case, the stress that we induced was every day, once a day, we were cleaning the tanks by lowering the water volume and brushing the side of the tanks. So we left the water down for about 2 minutes and then we brought it back up. In this experiment, we tested two feeds. Basal control diet, which contained moderate to low levels of good quality fish meal, 12.5%, and the rest of the ingredients, protein ingredients, are of plant origin. And then we tested the same feed, but further supplemented with 0.1% of the palatability enhancer. We did this experiment twice. So the first trial was performed in spring at water temperature, as indicated here, that is within the physiological optimum for the species. I'm also indicating the size of the fish at the start of the trial and the duration, which was 34 days during the undisturbed period and 6 days in the stress period. So what we can observe here is that when we don't have any disturbance and the temperature is optimum, we actually don't see any difference between the control and the palatability enhancer. However, as soon as we start stressing the animals, particularly the first day, we immediately see a decrease in the feed intake in relation to their pre-stress level, which is shown here by the bar. However, whereas in the control, we every day that we're applying stress, we see that the feed intake stays below the pre-stress level. When we supplement the feed with the palatability enhancer, we observe that quite quickly we resume feed intakes that are at the level of the pre-stress feed intake level. We're also seeing similar results here. Basically, this is showing the dark bars are the global feed intake, the average feed intake in the 6 days previous to the stress and the 6 days of the stress, just to have a comparable period of time. And we see a significant reduction of 29.5% in the control, which was mitigated with only 4% reduction and non-significant by the palatability enhancer. But then we repeated this experiment in winter. And as you can see here, the temperature in this case was below the physiological optimum of the species. Still, again, indicating the size of the animals and the duration. So 14 days in this case and still 6 days for the stress period. So in these more challenging conditions, particularly due to the low water temperature, even though the animals were undisturbed, in this case, we observed that we were able to significantly improve feed intake, which was slightly depressed in the control, by adding the palatability enhancer. And this was about 21% improvement. However, when we looked at the stress period, so both on a daily basis and also looking at the accumulated 6 days before and after the stress, what we observe is that in both cases, we had a significant reduction of feed intake. We have to understand that here we have two stressors, the management-related stress, such as the cleaning of the tanks, but also the low temperature. But what we do observe is that whereas the reduction is really extreme and significant. In the control, we have some days where the fish were not even eating at all. This was somewhat mitigated, and we had a significantly better feed intake during the stress period by the palatability enhancer. Again, sorry. Okay. Regarding the, so in the same period, in the same experiment, we were interested in also assessing whether some of the peptides which we commonly associate with the gut sensing mechanisms were also being affected. So whether the chemicals were also being sensed at a gut level. So in order to do that, in the first trial that were performed during spring, we kept the animals undisturbed for further 2 weeks, and then 24 hours after fasting, we've dissected different sections of the gastrointestinal tract. So in the stomach, what we observed was a massive upregulation of ghrelin in the palatability enhanced group. Ghrelin, as you know, is also known as the hunger hormone. And CCK, which is an anorexigenic peptide, meaning that it depresses appetite in feed intake, was quite clearly and significantly downregulated. So basically, these changes in gene expression are in line with the improved appetite in feed intake that we observe in this treatment. In the foregut or anterior intestine, although results were not significant, but there was a numerical trend for a higher expression of CCK. This could seem a little bit contradictory to what we observe in stomach, but it has to be said that CCK has different roles or functions. And of course, its main effect is going to depend on where it's being secreted, in which region of and also in the timing of its secretion. So CCK, besides being anorexigenic, also can affect digestion in a positive way by stimulating the secretion of pancreatic enzymes. And it also regulates intestinal peristalsis and gastric emptying, which affects gut transit rate. Then in the hindgut, we observed downregulation in the PE of PYY, which is a peptide that's similar to CCK, is also anorexigenic and also regulates gut transit rate. Now, just very briefly, I'm not going to get into the details, but just to say that we did a similar experiment in European sea bass, and this was actually just published very recently in the September issue of the Aquaculture Europe magazine. And in this case, our key findings were that, especially based on the period with no stress, we realized that sea bass is pickier than sea bream. So why do we say this? Basically because even under good temperatures of 21 degrees and undisturbed conditions, we managed to see 18% higher average daily feed intake in the palatability enhancer group, meaning that probably the moderate to low levels of fish meal that were used in this type of feeds were more of a challenge in terms of palatability for sea bass than they are for sea bream. In the case of the stress period, again, we had handling and low water temperature. We had a similar response as in sea bream, so a very large reduction of average daily feed intake by stress, which was significantly attenuated reduced by the palatability enhancer. And now I just want to share with you another example. In this case, looking at the utilization of a Luctamax type of formulation. If you remember, this is a powder product that is applied in the mash. So in this case, our objective was to see if we are able to improve the performance of rainbow trout when they are fed reduced fish meal diets. So here we have the conditions of the trial. Rainbow trout were 29 grams at the start of the trial. It lasted for 91 days. We had 3 tanks per treatment with 50 fish per tank. Another characteristic of this trial was that it was performed at a slightly higher density than what you would find in typical experimental trials, but in line with what you observe in commercial conditions. So we have here the density at the start and at the end of the trial. And of course, we were using good quality fresh water. So we had 3 experimental diets, a positive control with 15% fish meal and 5% of soy protein concentrate. In the negative control, fish meal was reduced to 7.5. Soy protein concentrate was removed from the diet, and this was compensated by poultry meal and feather meal hydrolysate, as well as by increasing a range of other plant-based ingredients. And then the third diet was the same negative control basal formulation, further supplemented with 0.25% of the functional palatability enhancer. So here are the results. In terms of growth, final body weight specific growth rate, we observed a significant reduction of growth in the negative control compared to the positive control, which was significantly improved by the supplementation with the palatability enhancer. Feed intake. I think it's interesting to show how this evolves over time. So what was clear, the effects were numerically always the same, and they were also significant, but the magnitude of these effects was reduced by the end of the trial, simply because, and this is also well known in fish, over time they will adapt both to the diet composition as well as to the higher density. But in any case, we always observed a significant reduction of feed intake in the negative control, which was partially mitigated and significantly improved by the functional palatability enhancer. FCR and protein efficiency ratio was not significantly affected, but numerically speaking, they are in line with what we observe in terms of growth and feed intake. So a worsening of feeding efficiency in the negative control, which is improved by the FPE. We also looked at digestibility, and maybe the most the most notable results were on the protein digestibility, as we would expect, keeping in mind what was the diet formulation. So there was a significant reduction in protein digestibility in the negative control, which was significantly improved by the FPE. And in this case, again, we looked at, in this case, only at the foregut to see some, to check on some of the markers of gut sensing mechanisms. And I'm only showing the ones that were showing more significant differences. So in the case of CCK, there was a significant upregulation in the FPE treatment. This is exactly, if you remember, exactly the same that we observed in CBRIM in the first trial. And as I explained previously, this could be influencing both satiety as well as digestion and gut transit rates. In this experiment, we also analyzed, which we didn't before, the expression of some taste receptors, in particularly the receptors from members of the taste receptor type 1 family, which includes 3 members, which associate 2 by 2 to form dimers, which are making then the umami and the sweet receptor. In this case, T1R1 was almost significantly upregulated in the FPE, and this is the dimer, the member that constitutes the umami receptor dimer, which is one of the main receptors that we are targeting with our formulations. Just want to say that very, very quickly to mention that both the results that I'm showing as well, other trials that were performed by us and other authors are suggesting or indicating that supplementation with this type of additives are in fact eliciting responses which are consistent with gut sensing mechanisms. And if you are interested in looking into a literature where further mechanistic evidences are shown, I recommend you to have a look at this publication. Okay, so now quickly to conclude, hopefully in this presentation you've seen how knowledge of taste science can be translated into practical applications to optimize aquafeeds. So by targeting the activation of fish senses, in particular taste receptors to sensory stimulation, we are able to improve the fish appetite and feed intake on one hand, but by activating also these receptors in the gut, we initiate a series of processes that can affect satiety, gut transit, digestion, absorption, metabolism, which ultimately improve feeding efficiency. So these effects overall then can also revert in a range of benefits, including gaining independence from marine ingredients, allowing a more efficient use of a bigger variety of ingredients, lowering feed wastage, lowering environmental contamination, and alleviate impacts of stress. So ultimately contributing towards a better performance, profitability, and sustainability in aquaculture. Now, last slide. So since I know that this is quite a new topic for most of us working in aquaculture, I've left you here with a list of literature which you can have a look at if you want to have a deeper dive into this topic. And of course, also feel free to send me an email if you want me to share some specific articles. And without further ado, I'd like to thank you all once again, and I'll be very happy to answer to your questions. Thank you.
Thank you very much, Sofia, for the fascinating and insightful presentation. I enjoyed it really much. And I think our audience as well. So we can see already some reactions. So great work, Sofia. You have given us a deep look into how feed palatability works from the fish perspective and how targeted scientific-driven solutions can have a real impact on performance, health, and sustainability in aquaculture. So now is the time to open up the Q&A. I would like to remind you all to send all the questions that you might be having for Sofia in the chat. We will start with the first question from Jorge Pino Marambio. Was the product applied to the top coating? Is the product expected to release in the water to increase attractiveness?
Yes, exactly. So, I mean, in the first trial, the product was applied by top coating together with the oil as vacuum coating. So the idea is that as soon as the feed is in the water, it can be quickly released and it will activate the sensory systems of the fish, so including both the olfactory receptors as well as the taste receptors, especially in the oral cavity in this case. So that is correct.
Thank you. The next question we got is from Stelios Tsekaros. What about the oxygen level of the trials, saturation levels in percentages?
Well, I don't have the data here, but of course, this is one of the parameters that is monitored throughout the daily in any experimental trial, and they were always within good levels. So it's not affecting in any way this parameter.
Mm-hmm. Thank you. The next question we got is from Neda Hilanayat. Hi, thank you for the very interesting presentation. What was the enhancer used in sea bream, the liquid coating? Then you expected this to improve the sensory impact or AAC, and how you could discriminate between these two mechanisms?
Okay, yes, this is a very good question. I mean, we have developed the two types of approaches, meaning a liquid product for where we believe we have a stronger impact on the sensory mechanisms, and a product that is applied in the mash, where we believe we have a stronger impact in the receptors along the gut, so targeting more the gut sensing mechanisms. But as I said, it's very, you obviously are having effects on both. As long as the chemicals are being released in the right places and in the right concentrations, and to activate, for example, sensory mechanisms, fish are particularly sensitive, so you need very, very low dosages. You are having this type of effect. So indeed, it's very difficult to discriminate where you are having the effect. But the way the products are designed is, if we really want to target feed intake when we have, for example, very acute challenges, on feed intake, then it's more important to have a sensory effect. Then we believe we will be more successful at that in having them at the surface. But the fish are incredibly fast eaters. So not all the chemicals are going to be dispersing into the water. So they will also reach the gastrointestinal tract. And the same when we apply them in the mash. In the end, these chemicals are also very highly water soluble. So even they are in the mash, a part of it will go into the water. So it can also be perceived by the olfactory and system and the taste system before entering into the gut. So honestly speaking, I believe we are targeting both, but we can try to prioritize one or the other depending on what is our main target and objective. So I hope this answers the question, because it's not an easy answer to give.
Well, if I think you answered really, really good. In case Neda has any other questions or sub-questions to this one, Neda, you can contact Sofia. Her email is mentioned on the last slide, so make sure that you note it. And any question you might be having, Sofia will be really happy to answer them. The next one, we can combine these two, I think. So from Pramod and Steen, how is the impact of the product in shrimp feed, and is affecting shrimps are comparable?
Okay. Well, again, a very interesting question here. Of course, we're talking the target was fish, basically because the chemosensory systems are totally different in these two species, as you would imagine, one being vertebrate, the other one being invertebrate. But I would say that we have extremely good results in shrimp as well, with similar type of formulations, because the underlying biological mechanisms are the same. So both fish and shrimp, even though they have different chemosensory systems, they rely on chemosensing using different types of receptors to locate at a long distance, find and finally evaluate their feeds. They have they all eat on similar type of especially carnivores. Let's say they will have they like certain type of ingredients which have a similar composition, high insoluble chemicals leaching into the water. So from that point of view, the same ideas behind how we develop this type of products are exactly translatable to shrimp, which we have indeed developed products for shrimp, and as I said, with very good results.
Thank you, Sofia. The next question we got is from Adela Aruayu. Hi, do you have any specific ways to analyze or check the leaching rate in the water of these additives?
Yes, not overall. We can, as I said, it's just a matter of putting them into the water and analyze chemically what is coming out. Of course, we are limited in a way by the technical capabilities. I mean, the detection threshold in water. But as long as we don't use huge volumes of water, we are still able to detect them. And actually, there's quite low threshold detection levels, as well as for the animal receptors themselves. So this is possible, of course, to assess.
Thank you, Sofia. The next one we got is from Jung Chul Kim. Thank you for the excellent presentation. I have one question regarding palatability enhancement. Is it possible to replace fish meal with plant-based protein sources by improving palatability alone without adding additional enzymes?
Without adding additional enzymes to improve palatability?
Yes.
Okay. Well, yes, of course, but I think we're talking about the enzymes in this aspect is affecting more how the digestibility of these plant materials, plant ingredients, which, as we have seen, with even the palatability enhancers through the gut sensing mechanism, we are able to somewhat improve the endogenous digestibility of these ingredients. But if we're thinking just of palatability, then this is even a clearer effect, because plant ingredients overall, they do not contain these substances. So soluble attractive chemicals. So by including this type of palatability enhancers, this is a way of replacing the attractability and palatability stimuli of marine ingredients and fish meal, including fish meal. So we have tested, it's true that we've never went down to 0% fish meal, except for in the case of shrimp. There we had very good results even with 0% fish meal. In the case of fish, to be honest, we didn't, because from a practical point of view, this is not being used in the industry. But definitely we can reduce fish meal to quite low levels and maintain a positive palatability in the diet. So actually, this is one of the main applications of this type of approach.
Mm-hmm. Thank you, Sofia. The next question we got is from... We got so many questions, so I lost a little bit the track. Okay, so the next one is from Leviet Dunk. So the fish have the same umami receptor to human. We can add glutamate into fish feed. Have you tried high levels of glutamate in fish diet?
Okay, yeah, very interesting question. I'm particularly grateful for this one, because there is quite a big bias when we talk about umami products or taste. We are quite conditioned by ourselves, humans, and it's true that the umami receptor in humans has very narrow activation, and it's almost only activated by glutamate, which makes sense because glutamate is the most important amino acid, or the one present in higher quantity in all proteins. So it's okay, I guess, that it's signaling the presence of protein. But actually, we humans are quite unique from that point of view. All other vertebrates, including fish, especially fish, but from swine and so on, and mouse, rats, and actually their umami receptor has a much broader activation profile. So actually they're activated by a range of amino acids, not only glutamate. And glutamic acid is one of the activators, but is not the main one, and especially not in fish. So in fish, there's one of the publications that is in the list, which has looked at, and actually there's more, that looked at what amino acids activate the umami receptor in different fish species, and glutamic acid is one of them, but is not even the strongest one having an effect. So I believe that in humans, yes, definitely glutamate is what mainly stimulates the umami taste, but in all other vertebrate species, it's better to have a combination of different amino acids rather than relying on glutamate.
Thank you, Sofia. The next question we got is from Pramod Kumar Singh. How about the thermostability in case of extruded fish feed?
Okay. So here, with respect to the tastants that we normally use, so the chemical compounds that we are using to target taste receptor activation. So in the powder, this is why we also this is our main target in the powder applications. They are completely thermostable, so absolutely no problem with extrusion and so on. In the end, these are chemical substances that are also present in feeds, in fish meal, having this effect, the same effect that we try to replicate with this type of chemicals, and we have no problem with extrusion. In the liquid formulations, where we are wanting to have a stronger effect on the senses, so here we are also including the olfactory system. So there we also use, depending on the formulations, but we also combine with aromas. Okay, the aroma is more the smell, usually of typical marine aromas, and there we have a problem with some of the chemicals that we use of thermostability, which is another reason why it's important to have it as a liquid formulation that is applied.
Sophia, I think I lost you. Yes.
Yes, I was away for a few seconds, but I think I'm back. Yeah. Okay.
Yes, I can hear you now.
Sorry about that.
No problem.
Did you finish my? Was I able to finish my response?
I'm not sure if there is anything to add to that.
No, then it's okay.
Okay. Perfect. So we are actually almost at the end of the Q&A time, but we received a lot of questions. So Sophia, do you mind maybe staying for 5 more minutes and answering on the questions?
Yes, no, I'm fine.
But at the end, we will not be able to cover all of them. So I will pick just a few. To the ones that we are not going to be able to give you an answer, can you please note that, can you please mark the Sophia's email? So please email her, and as I said, she will be really, really happy to give you an answer on them.
Yeah, for sure.
So the next one is from Alexander Yaramilo. A great presentation. Have you looked into feed ingredients that can reduce appetite or affect palatability in fish?
Yes. I mean, I think in aquaculture, we have 2 types of ingredients where we can have, let's say, reduced appetite or feed intake. Some are just the ones that are normally, they don't contain such high levels of, so it's more like the lack of chemostimulant substances, as I mentioned, mostly plant materials, as well as some of the novel ingredients. And the reason for that is just usually fish do not associate these with their natural food, so they don't really identify it as foods on one hand, and on the other hand, they don't have a lot of soluble chemical compounds. So these are more, they have more negative problems due to the lack of chemicals. But then we have another type of ingredients. Well, these are not so much ingredients, I would say, because I don't think that they would reach the level of being included in ingredients, because they can cause strong rejection problems. So more like certain additives or chemical compounds, which can be medications or certain types of functional additives like phytogenics, not all of them, of course, but some of them. And this can create a negative response because they are bitter. Okay? And the bitter taste is also a taste. There's many different taste receptors for bitter, which once they are activated, they will cause a strong aversion reaction. And this is basically innate protective or defense system that all vertebrates have to prevent them from ingesting something that could potentially be toxic for them. Basically because all plant metabolic secondary metabolites are, or many of these are bitter, and they can be harmful for the animals. So when we have this type of substances, then we can also have strong negative responses, and in a way is a more difficult challenge to solve than having simply the lack of these chemical substances. But also this type of approaches can be used, palatability enhancers can be used in a way to mask the presence of these chemicals.
Thank you, Sofia. The next question we got is from Ariana De Stefani. Thank you for your detailed presentation. I would like to know if there are any plans in the future to test the products in an aquaculture setup at sea. Thanks again.
Well, yes, of course. And actually, we've done that, but that's more through collaborations with directly with customers. So it's data that I cannot show publicly. But if anyone is interested in doing it, of course, and also we ourselves, it's not as easy to have trials in commercial setups, but definitely it will be interesting to do.
Thank you, Sofia. The next question we got is from Ingrid Sandabecken. Can you say anything about the raw materials used to produce these palatability enhancers? Fish byproducts, agriculture byproducts, or other sources?
Okay, yes. Well, actually, as you know, and you pointed out, marine byproducts are very often used, very commonly used as palatability enhancers. In our case, our products are quite different from that. Because we don't use any animal-based ingredients. So all our chemicals in our composition, they can be naturally obtained, but from non-animal, so meaning vegetable hydrolysates, yeast hydrolysates, and so on, as well as pure chemicals, which are obtained either from synthesis, so synthetic or purified. So in this way, we believe this is a distinguishing feature, is the fact that we are able, by using this type of ingredients, to maintain a much more constant and consistent composition compared to byproducts, which are, again, they can be quite efficient, but their composition, being animals at the end of the day, they will be varying across the year, especially depending on the time of the year, their biological body reserves, if they're reproducing, not reproducing. So it's much more difficult to control and homogenize the composition in that case.
Thank you, Sofia. We will only have one more question for today, and that is from Paulo Lozano Sulon. Thanks for the presentation. My question is, what correlation exists between attractant leaching and the feed acceptance rate?
Attractant leaching and feed acceptance rate. That's a good question, and to be quite honest, I don't have the answer out the top of my mind in fish, at least, because unfortunately, and I hope this will change in the near future. There are not so many people working on palatability in fish. As I said, I think this is going to change because we're clearly, as marine ingredients are being reduced, we need to have other solutions. So we need to understand palatability of ingredients, but we also need to understand the fish feeding behavior. So the question that you are posing is clearly associated with the feeding behavior. And there aren't a lot of methodologies that are standardized and that researchers are using to answer that question. For shrimp, yes. Shrimp, there is much more work having been done for many decades already on feed intake and behavior. And there's also a very interesting study. If you are interested, just drop me an email and I can share it with you. In shrimp, that is correlating quite closely the rate of leaching of the chemoattractive substances or chemostimulant substances with the rate of ingestion, how fast the shrimp is ingesting the feed. And actually it shows that below a certain threshold, when the pellet is not reaching such high concentration of these chemicals, the shrimp drops the pellet and they'll go looking for a fresher one. So there's a really nice, very cool study that was done by an American group on this. Hopefully more to come by fish in the future.
Thank you, Sofia. I would like to send you a big thanks because you presented an amazing presentation, and we could see quite like a good feedback from the audience, many questions that came. So thank you very much, and thank you very much to Lukta for being a corporate member of the ES. As we can see, reactions are just coming from the audience. Thank you very much for joining us today, and I wish you a lovely day ahead.
Thank you all. So thank you, Anna.